Good afternoon, everyone. We're gonna wait for 2-3 minutes and then we'll start. Okay, as there are yet we have more people who will be joining, so we'll wait for 3-4 minutes and then we'll begin. So I think we should begin now. So any problems in whatever we have discussed yes in last lecture, guys? Any problems?
No, sir.
So I think what we had discussed in last lecture was we were done with the, I was just seeing the lecture notes which I had sent you. We're done with the male reproductive system. Okay, we're done with the male reproductive system. Okay, so we will begin further. So there are two ways to go further. Okay, that either we can study male gametogenesis or we can study female reproductive system. As you wish, I can go along. Usually I take male gametogenesis and then we go on to female reproductive system and female gametogenesis, because once you learn the structures of male reproductive system, we should learn how gametogenesis or gamete formation happens. So I think we should proceed with that. That will be better for understanding purpose. Okay. So if you have any doubts in male reproductive system, you are free to ask, as we will proceed with the male gametogenesis, or what we call as spermatogenesis. Okay, so we'll proceed with that. Okay, so just a moment, I'm getting call from your school. Okay, excuse me. Just yesterday we'll have your lecture at 8:30 to 9:45, and then we'll have 11th lecture. Okay, so anyways, now we'll begin with this spermatogenesis. I hope all of you can speak and all of you have no problems in what we have covered in the last lecture. Okay, be communicative. Okay, otherwise there is no point. We will be at the loss. Otherwise I'll be doing my duty and that's all. Okay. So basically, when we talk about this spermatogenesis, okay, there are three major events which we discuss, and it begins in at puberty in the testes, as you all know. Let me share the board. Okay. Okay, so I hope you can see the board. So in today's lecture, we'll be covering the male spermatogenesis. It is one of the most important topics for questions, so kindly pay your attention. Okay, so we'll start with the male spermatogenesis. If you have any problems, you can ask in between. And okay, so the first thing is we're going to discuss what we call as obviously it will be in male, and you should know it begins at puberty. Okay, it begins at puberty. Okay, so the location, if we talk about specific location, in it is in obviously in the testis, but where? Anyone, can you tell me where it is? It will be in seminiferous tubules. Seminiferous tubules of testis. Okay, got it. So this is all you should know. And now we will talk about the entire process. Okay. So basically, predominantly, you can describe or you can classify this process into this manner, that first there will be spermatogonia, which is what we call as the immature germ cells. And from them, there will be production of the sperms. Okay. So there are three major events. So there is, first you should know about the spermatogonia, and then we can begin further. Okay, so spermatogonia are basically immature male germ cells. They are basically immature male germ cells. So we call them as immature male germ cells. So how are they? They are basically diploid. So they will have how many chromosomes? You know, human beings, male or female, diploid cell will have 46 chromosomes. Will have 46 chromosomes. Remember, it is all based on the chromosome. The chromosome numbers खास ख्याल रखो, बाकी confusion थશે. Okay. So they are present where location is in the inside wall of seminiferous tubules. Location is in the inside wall of seminiferous tubules. Okay. So this you should know. So basically, the first division which they will show is mitosis. They will show mitosis. Okay. So this starts from the spermatogonia. Okay. So if you take a single spermatogonian, okay, if I draw a single spermatogonian, it looks something like this. Okay, this is the how it looks. So it is diploid. Now there are three major events. Okay, there are three major events which happens in this spermatogenesis, which we are going to discuss. Okay. Remember the number of chromosomes and all, and they are diploid cells. So the entire process not given in your textbook completely, but nevertheless questions has been asked, so I have to teach in that manner. So there are three major events. First is formation of something termed as spermatids. Wait, I will explain what it is. Then there is something termed as permeagenesis. Okay, there is something termed as permeagenesis, and then there is spermiation, and then there is spermiation. Okay, so basically there is first is something termed as formation of the spermatids, then there is permeagenesis, and there is permeation. Okay, so what it is which we'll discuss. So this spermatid formation, it is basically a process to form something called spermatocytes. Okay, it is basically a process to form something called spermatocytes. Okay, then we have spermiogenesis, and then we have spermiation. So spermiogenesis mainly refers to formation of the sperms. Okay, spermatids are transformed into sperms. So in here, what will happen, that spermatids are transformed into sperms, and sperms are released. Okay, the release of sperms is what we call as permeation. The release of sperms is what we call as spermiation. While this describes the division of, these are formed from division of Spermatogonia division. Okay, so these are formed from what we call as the Spermatogonia division through via meiosis. Ideally, it is also a few more things, but as per your textbook, it is through meiosis. We could also express mitosis, which we'll discuss, and then we'll talk about this. Okay, so any problems in these three phases? Now we'll start with the first phase. We'll start with the first phase. Any problems, guys? Else we can go further. So first is spermatid formation. I'm assuming there are no problems. Kindly be responsive, guys, otherwise I won't enjoy. Okay, I feel that I am speaking to non-living things. I hope you all of you have brain and you are living things and your brain is working, so respond. So this spermatid, okay, formation, it has mainly three phases. That is not given again in your textbook. That is, first is multiplication phase. Then there is growth phase. And then there is maturation phase. So there are these three phases are there. Okay. Now here, this Spermatogonia will undergo mitotic division. Will undergo mitotic division. Okay. It will undergo mitotic division. It will form primary spermatocytes, which we'll talk about further. And this primary will basically be transformed into secondary. And okay. So the secondary spermatocytes, okay, are what we call as spermatids. So it will undergo mitotic division. Okay. I'll explain on diagram also. Wait. Now, and produce primary spermatocytes. Okay, these primary spermatocytes will grow okay, there will be DNA replication. Okay, so there will be mainly growth in them. Okay, so there will be increase in size and they will show DNA replication. Okay, such things will happen. Now this primary spermatocytes you can also write down as like this. Now over here this primary spermatocytes will undergo meiosis 1. Okay, will undergo meiosis 1 and then they will undergo meiosis 2. Then they'll undergo meiosis 2. So look over here. Over here there is first mitosis, then there is meiosis, and then there is meiosis 2. So what will happen? Let me explain. Okay, so let me show you. So what will happen? Let's say we start with the Spermatogonia. Okay, this Spermatogonia will undergo what? It will undergo mitosis. Okay, so mitosis means it will form. So this is 2n equal to 46. So this one will also have 46 chromosome. So now what it will form? It will form what we call as primary spermatocytes. So these are also 2n equal to 46. Got it? Now this will undergo meiosis. So see, if you look at this, we have formed primary spermatocytes by mitosis. Then they will undergo meiosis 1 and 2, as you see in the maturation phase. So now this will undergo what we call as meiosis 1 is a reduction division, as you know. Okay, so it will form secondary spermatocytes. Okay, it will form secondary spermatocytes after first, and then there will be. Okay, so there are two cells which will be formed and we will call them as okay. So from each of this there will be formation of, this was 2n, this is n, so it has reduction division 23 cells. So it has n equal to 23 chromosomes because it is first meiosis. Okay, then the secondary will undergo further division. So the secondary spermatocyte formation je thase ત્યાં સુધી it is considered as a what we call as the this condition, this secondary spermatocyte. Now this will undergo M2. Now there will be meiosis 2 and it will form what? It will form something termed as spermatids. So from each of these, through this again there are 4 spermatids are formed. N is equal to 23. Each one will have 23. So there are 4 spermatids. These are haploid, as you can understand. That meiosis 2, it will form this haploid condition. Okay, so we have this stage is all of this will be considered in the first phase, that is spermatid formation. That is the spermatid formation as we discussed. Okay, and over here we have spermatid formation, as you can also write it down. So any problems in this?
No, sir.
Okay. So what we have discussed is the first state, that is we are done with the formation of spermatids. We are done with the formation of spermatids. That is the first phase. Okay, we are done with. Now we are moving on to the second one, then we move to third one. Okay, so this is till this spermatid formation. It is very important. Okay, you should know the chromosome numbers also. At each stage you should know about the chromosome numbers. Okay, they ask these questions. Okay, primary is how many, it is diploid or haploid, so you should know spermatids, and secondary are haploid, rest of all are diploid. These are also diploid. Okay, now moving on further. Next is most important for questions. Now what I'm going to discuss, that is permeagenesis, or what we call as formation of sperm. It is most important. Is most important for NEET questions, anyone board. Okay, so now spermatids are formed. Now spermatids are formed. So what will happen now? Now this phase, this process will lead to formation of polarized spermatozoa. Okay. So there is the first thing is are formed. Now we'll wait. I'll explain. The sperm shape, which you know, it will be happening in this manner. So how this will go? How this will happen? So let us say this is the spermatid. Okay. Now this spermatid, there will be changes in the shape. Okay, this spermatid will become like this. Okay, there will be grow. This is nucleus. There will be flagella will be formed. Then this will assume this sort of shape. Okay. Then it will assume this sort of. If the meeting ends, join on the same ID. Okay, then just a moment. This is not pen and paper, so I have to. Okay, so in this manner, the sperm will change its shape. Okay. Okay. So this change in the shape is what we call as this process is what we call as formation of sperm. Okay. So first thing is over here, it will have a bigger nucleus. Now here if you see, nucleus starts to shrink. Okay, nucleus shrinks. As you know, this is nucleus. Now if you look at the nucleus, it is started to shrink. Okay, then there is formation of the acrosome. So this part will start to form. This part will be formed. Okay, so this is what we call as acrosome, which we'll talk about in sperm structure. The front part, acrosome starts to form. Okay, cytoplasm is lost. Okay, acrosome is formed from Golgi apparatus, cytoplasm is lost, centriole and mitochondria rearrangement will happen over here. This part is acrosome. As you can see, it is formed from Golgi body. It is formed from Golgi body. Okay. Then there is loss of cytoplasm. So all these changes will occur in the process of sperm formation or spermiogenesis. Okay, all these changes will occur in the process of sperm formation of spermiogenesis. Okay, mitochondria will be arranged over here. Centriole will be arranged over here. We'll discuss in this sperm structure. Okay, so what will happen? All this process, they ask these questions. Okay, that acrosome is formed from. The answer is Golgi body. It is formed from Golgi. Okay, centriole and mitochondria will arrange in the center as I have shown you. There is a rearrangement. Okay, and one more thing, that mitochondria will be arranged in a spiral shaped structure. Mitochondria will arrange in spiral arrangement. Okay, spiral structure. And then the covering of the sheath of mitochondria. Sheath or covering. This question was asked in 2017 NEET, not given in textbook, that the sheath surrounding mitochondria is termed as Nabengan. It is termed as the NEET question. Okay, the sheath will arrange in spiral form. It is also asked. So we can conclude that spermatids will differentiate into sperm by the help of permeogenic factors released from the cells which provide nutrition to sperms, called as Sertoli cells. Okay, and during differentiation it provides nourishment to sperm. Okay, so this is what we have discussed in this second phase. Okay, so usually sperms are embedded in Sertoli cells and they're released in this permeation. So what will happen once again in this process? The polarize means nucleus will be going to one of the ends. As you can see over here, it is going. So this nucleus will shrink, cytoplasm is lost, acrosome or head will like form, centriole and mitochondria will basically get arranged in this part. Okay, there is a rearrangement, cytoplasm is lost, acrosome starts to form. Mitochondria will be arranged in spiral shaped, okay, and the covering surrounding mitochondria is termed as nabenka. Okay, and then we have this, in this way differentiation will end, and Sertoli cells will provide nourishment to the sperms. Okay, so this is what happens in the process of spermiogenesis. Any problems, guys, in this? This is where, this is the covering, and mitochondria will be arranged like this. This is mitochondria. This is the covering. This which you see is mitochondria spiral. They are arranged in this manner. So any issues in this? This we have discussed the first phase, then we have moved on to the second condition, spermiogenesis, sperm formation. And ideally there will be, this is, if let's say this is the Sertoli cell. Okay, if this is the Sertoli cell, then the sperm head, okay, will be embedded in it as it provides nutrition. So this is, let's say, Sertoli cell, and this is the sperm head embedded in it. So this is what you see is sperm. Got it? So any questions in this, anyone?
No, sir.
Okay. Just go through it once. Meeting will be end in minutes. So again, join on the same ID. Okay. So join on the same ID. This is second part gets over. Spermatogenesis. We'll start with the third part, that is release of sperms or spermiation.